this session is called Redefining Human Frontiers in Biotechnology and Reproduction. And we have 3 very interesting talks to look forward to, 20 minutes each, by first Hank Greely, Professor of Law from Stanford University, then Stephen Wilkinson, Professor of Bioethics from the University of Lancaster, and last Françoise Baylis, Professor of Bioethics too from Dalhousie University in Canada. So without further ado, I welcome to the stage first Professor Hank Greely. Most welcome, please.
Gratis på födelsedagen smer. I did that in part because I want to express my deep appreciation for all of you for being willing to speak in and listen in my language. My total Swedish vocabulary, I think, is 3 words: he, talk, and öl. If I learn those 3 words anywhere in the world, I usually can survive. What I want to talk about today is what my friend John Evans, a great sociologist who's interested in these areas, has said. What it is to be human is perhaps, in the academic world, the deepest of all morasses. And I agree with that. But despite that, that's what I'm going to be talking about, because I think the technologies are leaving us no choice but to explore that question more deeply. I have framed the question in almost a grammatical sense: human as noun versus human as adjective. I am a human. I believe, I'm informed and believe, and on that basis allege, that all of you are also humans. I have human hair, I have human DNA, I have human blood. That's an adjective. A human is a noun. It's a marker, it's a distinction between, one might want to call it person or not person, but being a human is different from being something that has some connection with humans in ways that I think are relevant to how we deal with them. Just about 5 years ago, Bartha Knoppers, a wonderful, now retired Canadian lawyer bioethicist, convinced me to write with her a piece for Science that was published in December of 2020 called Biotechnologies Nibbling at the Human, where the argument was that where biotechnology was taking us was going to force us to make some fundamental changes to what we think about when we think about what is a human. That was sort of an odd pairing because Bartha was more attracted to that idea than I was. So, we had the sails out and the anchor dragging at the same time, and I was definitely the anchor on that. But I think over the last 5 years, I've pulled the anchor up because I think things are moving very quickly. What I want to do in my short period of time here is to basically talk about 2 things. First, I'm going to talk about 4 technologies that are causing us to have to rethink this difference between human as noun and human as adjective. And then second, I want to suggest, at least tentatively, some possible ways for us to think about making those distinctions and deciding what we want to do with these various technologies. So the 4 technologies are about beginnings, endings, parts, and modifications. Beginnings. We've had to worry about when life begins for a long time, particularly for the last 47 years and 3.5 months since the birth of Louise Brown, the first test tube, so-called test tube baby. And with Louise Brown came human embryos, ex vivo embryos. And with ex vivo embryos came the questions that continue to plague us about what we can and can't or should and shouldn't do with embryos. I'm going to talk about 3 things with respect to beginnings. First is that old question of what we should and shouldn't do with actual embryos. Starting in 1984, there has been a convention, sometimes put into statutory form, that human embryos should not be used for research past 14 developmental days. Not counting days spent in the freezer, But 14 days of development, then you've got to stop. Coming from a British commission initially, commission's recommendation. This was an easy recommendation to give at the time, because nobody knew how to keep them alive for even 7 or 8 days. 14 days, it's always easy for scientists to give up doing something that they can't do. However, it's also not uncommon for scientists to later figure out that they can do what they thought they couldn't do. And starting about 8 or 9 years ago, scientists began to run up against that 14-day limit. And the question was, should we go beyond it? The International Society for Stem Cell Research in 2021 published new guidelines that said, yes, we should reconsider the 14-day rule, but they didn't propose a 21-day rule or a 28-day rule or a 3-month rule. They just said we should reconsider it. Actually, Françoise and I, along with Josie Johnston, published a letter to the editor in Nature saying, this is a serious mistake. The three of us didn't necessarily agree on what a limit should be, but we thought there should be some sort of limit. And now we're pushing even farther to getting past the potential, and it's been done with non-human primates. So far, I think everyone is adhering to the 14-day rule still with respect to humans, but for how much longer? And to me, and part of what drives me, if you could, which we cannot yet, keep a human embryo alive ex vivo until 8 weeks, when it's no longer called an embryo, but magically is transformed into a fetus, at least terminologically, or 12 weeks, or 16 weeks, or 22 weeks, at what point can you stop doing research on it? I think that's a question we're going to have to answer. Second complication is now we're talking about things that aren't just old-fashioned egg meets sperm. They shake hands and make an embryo. We're talking about embryo models, or embryoids, or synthoids, or gastroloids, many, many different names for them. But the idea is you're taking cells, stem cells, you're putting them together, you're giving them the right pushes and shoves, and they start acting like embryos. And they start dividing, and they start growing, and they start developing. And they start developing in ways that look an awful lot like the way real embryos develop for the first 6 or 7 days, and then as far as we can tell, for the first 12 or 13 days. But some of these have been taken past 14 days, though it's unclear. Should they be? Do laws that ban research on human embryos after 14 days apply to these things, which aren't embryos, at least in the old-fashioned sense? Do ethical guidelines apply? Should they apply? That's a question we're going to have to resolve. And then third, and this will overlap with some of what I think Stephen's going to talk about, there's interest in partial sort of end-stage ectogenesis. So making babies outside of people's bodies. We've actually been doing that since Louise Brown. She spent her first 7 days outside of a body, and there have been about 12 to 14 million humans born since then who spent their first few days outside of a body. In a sense, we've been doing it at the other end for many, many years through incubators and other things that will keep babies who are too premature to live on their own alive. There's a technology that was first introduced with sheep, or first research was done with sheep, called the biobag that we're going to hear about. If you did that with humans and you took a newborn who, say, was born at 20 weeks, too premature to survive. Right now at 22 weeks, it's iffy, 20 weeks, no chance. And you did this to extend ectogenesis back from the 40 weeks, 39 weeks, back a few weeks earlier. What is that thing? There are some people who say that thing is a baby. But other people are arguing for new terminology. No, it's a fetonate, or the one that really grates on me, a gestateling. A thing from gestation. So are these babies? Are they something short of babies? Are they fetuses when they're on the outside? What are they? We're going to have to answer that question. Let's go from beginnings to ends. We've been dealing with end-of-life issues for a long time, certainly since the late 1960s with the invention of brain death and the maybe not coincidental need for organs for transplantation. We have been dealing with when does life end? Well, that's getting more complicated in some respects too, in 3 respects at least. First, there's increasing use of what's called normothermic regional perfusion in organ transplantation. Now that's a long term. Normothermic just means blood temperature, body temperature. Regional means not the entire body, part of the body. And perfusion means running a fluid through it. And in this case, it's running blood through it. Now, normally for organ transplantation, you wait until a patient has been declared dead, a human, noun, has been turned into an adjective, a human body, and then you take the organs out. A problem is if you wait too long, the organs are less useful, et cetera, et cetera. There are 2 ways to declare people dead, 2 biological ways: total and permanent cessation of heart and lung function, and total and permanent cessation of all functions of the brain. There is a 3rd way we lawyers like. You can just go missing for 7 or 8 years, and after a certain period of time, you're declared legally dead, which is the basis of a number of very funny Hollywood movies from the 1930s and 1940s when the dead spouse shows up on the eve of the widow's or widower's next marriage. Well, with normothermic regional perfusion, the patient is declared dead on cessation of heartbeat, heart function, for 5 minutes. Not going to restart naturally. Then they go in, they attach tubes to the blood system, but they tie off the arteries leading to the brain. And they put blood flow at body temperature back into that human body, into that corpse, in a way that keeps the rest of the body alive, keeps those organs in good shape for transplantation, but prevents that blood from getting to the brain, where it could potentially keep the brain alive. And if the brain is alive, it makes it harder to say that this is that this is no longer the noun, a human, but is now a human body. This has become quite a controversy in organ transplantation. Second issue, what do we do with those human bodies that are being maintained physiologically alive, even though they've been declared brain dead? This is a little different from the normothermic regional perfusion for organ transplantation, where they've been declared dead by heart standards. This is dead by brain standards. It turns out we've known that you can keep those bodies alive for a long time. I think the current record holder is 30 years, somewhere in either the Persian or the Arabian Gulf, depending on which side of the water you're on. People have given birth several months after being dead. People have gone through puberty several months after being dead. Their bodies are alive, they're legally dead, and their brains are no longer functioning at all. Well, I've been looking for a good term for that, and I'm currently working on thinking about animated corpses as things that are corpses, but they're, it's Dracula. It's an animated corpse. Maybe it's a zombie. Recently, there's been an increasing new use of these animated corpses as research subjects, objects, things. People have been putting particularly pig organs, genetically modified pig organs for xenotransplantation, onto or into animated corpses to test them. Is that right? Now, this is with the consent of either the dead person before he died or the family afterwards. But should we be able to do that? Or if we can do that, why can't we just take them into medical schools and do an anatomy class on them? Certainly, it would be more realistic if you're training to be a surgeon, if you're cutting into a body that has blood flowing and lungs working, than in a body that's been fixed in formalin for days, months, years. What would be the limits of what we can do with animated corpses? And then the third one could fall into my next category, but I'm going to put it here because I think it's ickier here. Back in 2019, a Yale scientist named Nanad Sistan published a remarkable paper. He had gone to the local New Haven slaughterhouse and said, "Give me pig heads. Pigs you're killing for bacon and pork chops and so on. Decapitate them after they've been humanely killed, hang the heads on a hook, drain them for 4 hours, give them to my lab. They went to his lab. He ran a normothermic perfusate through them, not blood. He was using an acellular perfusate. And although he's a careful man, he didn't say the brains came back to life. He said the brain cells came back to life, which is quite remarkable because conventional wisdom has always been 10 minutes without oxygen, brain cells, central nervous system neurons are gone. Maybe if you're a little kid and you drown in a very cold lake, maybe you've got 20 or 30 minutes, but 4 hours, no way. Well, what if this works? It worked with the pig brains. He hasn't been doing it on human brains. What if you take the brain of somebody who has died, and you hook it up to this, and the brain stays alive? What is that? Is that a human brain, or is that a human? Now, it's kind of a locked-in human, but we've got experience with locked-in humans, people with ALS, or people with quadriplegia, or people with a variety of nasty syndromes who can't communicate or react. What are we going to do with those? Body parts. So you heard a little bit about organoids. I've actually done a lot of work on human brain organoids. They're not very worrisome yet. There may be 1, 2, 3 million neurons compared to the 89 billion neurons in our brains. But they're going to get bigger. And the guy I work with most, Sergiu Pașca at Stanford, has formed what he calls assembloids. I think of them as pearls along a string, where he connects a bunch of 2 or 3 million neuron organoids to each other, making something that's bigger. You get to 60 million neurons, you're the size of a mouse brain. Even at 1 million neurons, you're the size of a honeybee brain. And honeybees have some pretty sophisticated behaviors. These are human cells that are forming human brain-like things that are quite useful for research. At what point do we have to worry about, are they feeling pain? Are they conscious? Are they human? So we've got that. We also have with body parts something called bodyoids. Now, this was not my term for it, although I'm a co-author on a paper that talks about them. There is a crazy, I shouldn't say that, there is an ambitious and aggressive stem cell scientist at Stanford who came up with the idea of, let's build things, mice, rats, people, that don't have heads, but are alive. Or maybe they've got heads, but they don't have brains. Or maybe they've got brains, but they don't have enough brains to feel pain, to be conscious, to be sentient at all. Why does he think we should do this? Well, for mice and rats, so that you'll have laboratory animals that won't suffer. For maybe cattle and sheep, so you can eat your meat without worrying about animal suffering involved. For humans, maybe so we can have a store of organs for transplant. Or maybe humans for research purposes. Is a human body without a head that's alive a human? Or is it a human body without a head? So, modifications, chimeras, things that are part human and part non-human. I've been dealing with this for 20 years with respect, actually I think one of my first things was responding to a piece that Françoise wrote along with Jason Robert about human, non-human chimeras, putting human brain tissue into rodents. My friend Sergiu has now gotten rats, 25% of whose cerebrums are human cells, human brain tissue from human organoids. At that point, does the rat become humanish? And if so, what would that mean? We're also moving in the other direction, taking non-human body parts and putting them into humans, particularly these genetically modified pig hearts, kidneys, livers, for the good purpose of keeping people alive. But do they become part non-human as a result? Where do we start? Where do we draw lines with respect to these body parts? And the modifications. This is sort of now in the modification. We're modifying a human or a rat in directions that begin to blur the differences. And then we've also got questions of surgical modifications, of changing people. I don't know whether anybody's ever seen a photo of the lizard man who's changed his face to make it look very much like a lizard. These bod mod peoples are very excited about doing many things. At what point do we say, okay, yeah, you're a lizard. You're a lizard with a human body, but you're a lizard face. Some of these modifications, which are viewed as enhancements, may raise those sorts of questions. There are another 7 or 8 things I could talk about, except I can't, because I'm out of time, almost. But I want to say how to move forward. Some of you will have noticed a human adjective that I have not used in combination, human dignity. I don't believe in human dignity. I believe in the dignity of humans, but I don't believe in the dignity of our species. That strikes me as an argument people make when they don't have another, when there's something they don't like and they don't have another good argument to make against it. So you could say it's against human dignity, but what else could you say to try to decide how should we treat these? How much respect are they due? And I suggest we could start by thinking about what is it we care about? Was it born alive from a person? Does it have intelligence? Does it have consciousness? Can it feel pain? Can it suffer? Does it have sentience? Does it have human DNA? Is it, if we're using body parts, were they used with consent? And what kind of consent? And how much? And how deep? And how specific consent? There are a bunch of different questions about what we care about. Take those questions almost as a checklist. This is sort of a dumb idea, a Stone Age idea. Take a checklist and look at them against these new technologies in the context of different cultures. Different cultures are going to have different answers. Different people will have different answers. That's okay. It's a big world, and not everybody has to do the same thing everybody else does. Ultimately, I've come around to agreeing with Bartha that this is a bigger deal than I thought it was 5 years ago. But that paper also reminds me of something else. That paper was published on December 20th, 2019, almost 6 years ago now, at which point something was happening in Wuhan that we didn't know about that was going to change the world in ways we had no expectations of. So all of horizon scanning is a wonderful thing, as long as you remember that unlike the Earth's horizon, the horizon of the future often changes in unpredictable ways. Thank you for your time and attention.